Good afternoon, everybody. If you could please take your seats for the start of the next session. So I'm delighted to be able to start off the afternoon session. I'm Ananya Chowdhury and I'm Professor of Clinical Oncology and a clinician at the Christie in Manchester, specialising in bladder cancer. We've got 3 excellent, potentially prize-winning talks in this next session. So to start off, I'd like to welcome Simon Baker, who's going to talk to us about insights from in vitro studies to do with the BK virus as a likely risk factor for bladder cancer. Simon?
Thank you, Ananya. Hopefully you can hear me okay.
Excellent. Thank you, Simon.
Yeah, well, thank you very much for the invitation to speak. I'm really sorry that I wasn't able to travel down to London today. A family illness has kept me away, unfortunately. The organisers asked us to think about what were the big questions in bladder cancer. And for me, the biggest question in bladder cancer remains, what causes it? And the reason that's such a big question is that nobody has a really good answer for this question. And my work seeks to use mutational signatures to help us understand the causes of bladder cancer. Now, recently we were able to capture mutational signatures in the aging urothelium. If you're not used to looking at mutational signatures, this graph over here is called a mutograph. And you can see mutations are captured across the whole genome, and they're classified into 6 groups based on the type of base change that we see. And then they're further broken down based on the local context. So that's the flanking bases either side of the mutated base. And the reason that's important is that local chemistry really affects mutation. And if you're not used to looking at mutagraphs, then the main thing to concentrate is just the kind of pattern of the spikes and the colors where they're occurring for now. So this is a really recent description, happened only last year, and used a new method, NanoSeq, which allows us to look in normal tissues and was pioneered by the Sanger Institute. So smoking, we know, is the strongest risk factor for bladder cancer. And we generated this mutational signature in 2020 based on exposing normal urothelial cells to a smoke carcinogen. And you can see this really characteristic pattern of light blue peaks here. And these are exactly what we see in smoke-exposed cancers like lung cancer. There are some chemical exposures that we know cause bladder cancer. So in herbal remedies and some teas, we see aristolochic acid, and this creates this kind of distinctive pattern of T2A changes that we see over here. And what about infections? Well, we know some cause bladder cancer. So schistosomiasis in Africa is a risk factor for bladder cancer. Many of us would have expected recurrent UTIs to be important for bladder cancer risk, but that doesn't seem to be the case. But we know very little about viruses. And viruses are an important potential cause of bladder cancer because they induce the antiviral ApoBec enzymes. And these are cytosine deaminase enzymes that cause the distinctive patterns of changes that we see in bladder cancers. You can see that pattern here. It has this very distinctive periodicity. So you can see it's much bigger in the black and red sections and much lower in the blue. And the important thing to note is that, you know, this isn't a normal feature of aging. All of us aren't developing ApoBec signatures in our urothelium. And furthermore, exposing urothelium to smoke carcinogens doesn't induce ApoBec signatures. So what does induce an ApoBec signature in the urothelium? Well, I'm interested in BK polyomavirus, and it's a childhood infection. It infects about 80% of us, and you can see that here from the seroprevalence of antibodies against BK. But when it infects us in childhood, it causes cold-like symptoms, but then it becomes latent. It hides in our kidneys, and it can become reactivated in later life under immune-limited situations. So that might be immunosuppression for a kidney transplant, or maybe even just aging as our immune system declines. You can see here in this graph the increasing frequency of BK viruria with age. And BK is in the black bar, and there's another polyoma virus here in the gray bar showing that there may be other viruses involved too. Now, studies by Rick Bryan's group and others have shown that viral sequences are incredibly rare in the final tumors. So if a virus is going to be the cause of bladder cancer, it needs to be a hit and run carcinogenic mechanism, meaning it needs to induce changes that lead to cancer, but then it needs to be removed from the urothelium, and then the cancer can develop on its own without the virus. And the way that we study this is using normal human urothelial cell tissue models. So we take bits of bladder tissue and bits of ureter tissue, we strip the urothelium off, we put them into culture. So this is just a pure epithelial culture. We expand them in number, and then we redifferentiate them into a tissue that looks and functions like the normal tissue. Importantly, it's mitotically quiescent, but it also develops a functional barrier. So it's a tight resistive barrier. So what we did was to study those tissues infected with BK virus in the lab. And this mechanism here, we just published a few weeks ago in Oncogene. And what we think is happening is that in patients, a reactivated BK infection in the kidney leads to sloughing of epithelial cells. So these are proximal tubular epithelial cells containing the virus. The virus then infects the quiescent urothelium, and there viral large T antigen inhibits retinoblastoma and it inhibits p53. And this drives that mitotically quiescent urothelium back into the cell cycle. But these aren't truly proliferative cells. They become arrested at the G2M checkpoint. And they become arrested there because they've got DNA damage. We don't know for sure, but this is likely to be at least partly Apobec-induced DNA damage. What happens then is that the Apobecs, there's a war going on in these cells between the virus and the host. And the host unleashes these Apobec enzymes in the hope of damaging the viral genome and limiting the infection. But what actually can also happen as a side effect is that the host genome gets damaged. The Apobecs randomly damage the genome, and by chance, some cells will get damaged in driver genes in an activating fashion. The immune system then clears the virus, and these cells harboring driver mutations then will outgrow into tumors slowly over a process of years. When we looked in detail, we can see specific inductions of both Apobec3A. There are two products of the Apobec3A gene because there's an internal start site. And we also saw a stronger induction of Apobec3B. It's the lower band here. The upper band is 3G. So these Apobecs are a product of gene duplication, and they're very similar. So we knew the protein was there, but we needed to look for activity. And we used the kind of deaminase activity assay. And the assay mirrors the process that's happening in the body. So the Apobec deaminates the cytosine, so converts it into a uracil. And then the UNG, the uracil DNA glycosylase enzyme, removes the uracil, leaving an A-basic site. And then we can put the probe into alkaline conditions, and that cleaves the probe and makes it short. And we can detect this cleavage based on scanning to detect a fluorescent dye. And what we found was there was a strong increase in activity of Apobecs in our infected cell cultures. We could detect that by the increase in the presence of this cleaved band. And on the side here are negative and positive controls, respectively. So in the same way as was happening in our deaminase assay, in the host genome, Apobecs are turning cytosines into uracils, and then repair requires their excision by the uracil DNA glycosylase again. This is encoded by the UNG gene, and you can see that here was induced by infection. And it leaves an A-basic site, or here we refer to it as an apurinic, apyrimidinic site, suitable for later repair. And we found again that the host genome was damaged. These AP sites are increased in the genome by infection. So we can show that viral infection induces Apobecs, and that damages the host genome. which ApoBec is the most important is an open question. So we found a weak association for A3A with viral gene expression, deaminase activity, and those AP sites. But we found a strong and significant association for ApoBec3B with all of them. So at the moment, our data supports A3B as the major mutagenic ApoBec in viral infections in the urothelium. So here's the association of ApoBec3B with large T antigen, with the deaminase assay, and with those AP sites. So to conclude, there's etiological evidence from other groups linking polyomaviruses with the aging urinary tract and also with cancer risk that I haven't really talked about so much. Our in vitro infection model suggests an ApoBec-mediated hit-and-run model of carcinogenesis is possible, but we need more detail on the mutational processes and the tissue-specific signatures to build confidence that this is really a mechanism that underpins the risk of bladder cancer. And the way we're going to do that is to look for evidence in the renal transplant population. So I need your help, particularly if you're a urologist and you see a patient with urothelial carcinoma following a renal transplant. I'd really like to enroll them in our study. And what we need is fresh frozen carcinoma biopsy and a blood sample so that we can generate these high resolution mutational signatures and really put BK on the map as a serious risk factor for bladder cancer. Leaves me just to say thank you to my collaborators. All the work I've reported today was funded by York Against Cancer, our local charity, and my work going forward with mutational signatures will be funded by a Kidney Research UK fellowship. Thank you.
Thank you very much, Simon. Do we have any questions? We've probably got time for one or two. So, Simon, I'm going to kick off if that's okay. Given it sounds a bit like this virus is quite prevalent in young people, what sort of predisposing factors could there be for this to cause, I guess, carcinogenesis in some people, but not in others?
Yeah, so it's thought that 80% of us have the virus living inside our systems, but it's kept suppressed by our immune systems. So the susceptibility risk factors would be those relating to re-emergence of the infection from its latent phase in the kidney. And that's really poorly studied partly because re-emergence of the viral infection is asymptomatic. So because people with a somewhat infective immune system wouldn't have complete urothelial damage like we see, for example, in a bone marrow transplant patient where they have extreme immunosuppression. They have BK infections that cause a gross hematuria because they cause hemorrhagic cystitis. So basically, in that situation, the virus infection is so florid, it destroys the whole urothelium. Now, in most people, we have kind of a decently functioning immune system that might just be compromised by a different viral infection, for example, or just the limitations of aging. And so the risk factors are going to be what's kind of limiting your immune system's ability to suppress the reactivation of the virus in the kidney.
Okay, thank you.
Hi, Simon. Do you think this effect is simply the hit and run, or is there continued ongoing high levels of ApoBec activity once you've developed the tumour? I say this because I believe I'm right in saying that the TCGA showed that in tumours with a higher ApoBec signature, there was higher expression of ApoBec3B.
Yeah. No, Doug, that's absolutely right. So in a number of different cancers, we see ongoing ApoBec activity in the tumor itself. And there are a number of different ways of regulating ApoBec activity. For example, PKC activity can induce ApoBec activity. So there could be lots of non-viral triggers for ApoBecs. And so this is one of the real tricky things about pinning the ApoBec damage on the virus. So what we need to be very careful about is refining the detail of the ApoBec signature that we get. That's why these fresh frozen samples are so critical, but also relating it into the tissue-specific transcriptome and epigenome at the time of infection. And that will tell us where we would expect to see these signatures damaging the genome. So we're trying to get into some really kind of high-end bioinformatic analysis integrating multiple omics to really pin the damage that's initiating on the virus. We certainly don't think the virus is there in the final tumor. And it's possible, though, that tumors can become reinfected because, there's no reason that somebody bearing a cancer wouldn't have BK reactivation during that process. But we don't think that the virus persists right from initiation into the final tumor, except in the very rare cases of some renal transplant patients where they have viral genome integration into the host. But that's definitely not a feature of carcinogenesis in the immunocompetent.
Thank you very much. So in the interest of time, we're going to move on to our next speaker. I'm delighted to invite Chi Kin Ten to the stage to tell us about gut microbiota and radiotherapy for bladder cancer.
Good afternoon, everyone. I'm Chi from Professor NQT's lab. My research question is, can the gut microbiota both radiosensitize tumors and spare normal tissue toxicity? Actually, in this relatively new world, we are still sailing across the ocean of gut microbiota, looking for highly valuable bacteria, which can bring benefits to human health, to cure disease, or in our case, to improve treatment response. Our lab is interested in radiosensitization. Previous lab members have proven that HDAC inhibitors, histone deacetylase inhibitor, can radiosensitize the bladder tumor, which means HDAC inhibition is a promising mechanism of radiosensitization. So what about an endogenous radiosensitizer with fewer side effects? One group of such inhibitors are short-chain fatty acid. So an interesting phenomenon is that the gut microbiota can ferment fiber to produce short-chain fatty acid such as acetate, propionate, and butyrate, which can accumulate in the cancer cell to a sufficient level of HDAC inhibition because of Warburg effect, so-called aerobic glycolysis. A paper in 2016 have also proven that high fiber diets can delay the tumor growth and also increase one of the short-chain fatty acid, butyrate, in both the plasma and tumors. So that's why we are thinking if we can treat the mice with high dietary fiber, leads to a modified gut microbiota, enhanced production of short-chain fatty acids, accumulate in the tumor cells, make them be more sensitive to radiation. So this is the first part of my project. I did it in immunocompromised mice. So as we can see, each dot represents the gut microbiota of a mouse, and dietary fiber in this shape them to a unique profile because we can see the gut microbiota profile within the same group clustered with each other and rather different from the others. So after receiving ionizing radiation, here are different dietary groups. High soluble fiber, inulin, had the best tumor response rate. However, it was interesting at the time that only 4 of them responded to the radiation, but 4 of them did not. So I further looked at their individual tumor growth curve and further separated them to responder and non-responder. And in their gut microbiota, I realized that responder had almost 20 to 35% of Bacteroides acidificans, and non-responder had none of them. And also by using correlation analysis, I saw that the responses are influenced by gut microbiota. Since we know that the gut microbiota, dietary fiber, radiotherapy can modulate the anti-tumor immune response, so I have to move our experiment to immunocompetent mice. So that's why I injected mouse bladder cancer cells to black 6 mice, and at the same time, I started them with low fiber diet, which is 0.2% cellulose, and high fiber diet, which is psyllium. Psyllium is fibrogel used to mitigate the side effect for radiotherapy patient. It can increase water content of feces, which may help facilitate the bacterial fermentation. Psyllium plus resistant starch, a well-known butyrate, short-chain fatty acid producing fiber. Psyllium plus inulin, a potential radiosensor found in the previous project. And after that, there are 2 cohorts. One cohort, I just monitor the tumor volume until 100 cubic millimeter. And for the second cohort, I gave them 6-gray radiation and monitored them up to 700 cubic millimeter. So for the first cohort, to study the effect of dietary fiber alone, I can see, so orange is the low fiber diets, red, blue, and green are the high dietary fiber group. As we can see, psyllium plus resistant starch and psyllium plus inulin can significantly delay the tumor growth. And also, the gut microbiota and dietary fiber may execute their function via secretion of metabolites, So that's why I did untargeted metabolomic analysis of the secum content. And I can see psyllium plus resistant starch increase the butyric acid, and then psyllium plus resistant starch increase the inosine level. However, I knew nothing about inosine at that time. That's why I Googled it, and I realized that there were 2 papers published in Science and Nature Metabolism in 2020 have proven that gut's microbiota produce inosine can enhance the efficacy of immune checkpoint blockage via activation of cytotoxic T cells. So this reminded me to look back at the local tumor immune response. And I found that psyllium plus inulin can increase cytotoxic T cells, CD8 cells in the local tumor, but interestingly, it will decrease in psyllium plus resistant starch. And I got a consistent result from nanostring platform among all different immune cells. CD8 cells, marker of cytotoxic T cells, was increased in psyllium plus inulin, but it was decreased in psyllium plus resistant starch. So it seems like psyllium plus inulin and resistant starch, they might achieve tumor control via different mechanism. Immunomodulation might be involved in psyllium plus inulin. So in the second cohort, to study the radiosensitizing effect of the fiber, when the tumor grew until 100 cubic millimeter, I gave them 6-gray ionizing radiation and monitored them out to 700 cubic millimeter. So this is the result, a bit complicated, but please follow me step by step. So all the solid lines are non-IR control, and the dots line are radiated mice. So again, orange is low fiber diet, and red, blue, and green are high fiber diet. So from this radiated cohort, we can see psyllium plus inulin and psyllium plus resistant starch can significantly delay the tumor growth combined with ionizing radiation compared to 0.2% cellulose and also psyllium, which is promising. And also interestingly, psyllium plus resistant starch in this cohort had even had a better effect compared to psyllium plus inulin. So I further used the time for the tumor to quadruple in volume as an endpoint for this survival analysis. I can see, so in each dietary group, 0.2% cellulose, psyllium plus resistant starch, and psyllium plus inulin, ionizing radiation can improve the survival, especially in psyllium plus resistant starch and also psyllium plus inulin. So to study whether the tumor response was affected by the gut's microbiota, so I also analyzed the gut's microbiota. Again, we can see the gut's microbiota within each dietary group cluster with each other and rather different from the others, which means that we modified the gut's microbiota again. And when correlate the gut's microbiota with the tumor growth, we can see Bacteroides species and Clostridial order negatively correlated to the tumor growth, which means the more this bacteria in psyllium plus resistant starch or inulin, the slower the tumor growth. And also interestingly, as we can see, psyllium plus inulin can enhance the local tumor immune response. And also this bacteria in psyllium plus inulin also positively correlated with the systemic cytotoxic T cells as well. So which means that this is an association between the gut's microbiota and the enhanced immune response. So again, to look at the local tumor immune response, so this is the immune response for the second cohort. As we still can see, cytotoxic T cells will enhance in psyllium plus inulin, but it will decrease in psyllium plus resistant starch, which is consistent with the first cohort without IR. And also, I also did some plasma cytokine assay and FACS analysis of the spleen. I can see both of these cytokine and cytotoxic T cells were negatively correlated with the tumor growth, which means the more these cytokine or immune cells, the slower the tumor growth. So, I mean, for the tumor response, I have shown that psyllium plus resistant starch and inulin that can combine with ionizing radiation that can delay the tumor growth, and then it is associated with metabolites production and also immune response. So during the radiotherapy, we always hope to increase the tumor response but decrease the side effect. So I would also like to know whether the gut dietary fiber and gut's microbiota can protect the guts from radiation-caused injury or not. So that's why I also treat the mice with dietary fiber for 2 weeks, and then I gave them radiation to the lower abdomen, centered on the urinary bladder. And then 3.75 days and 24 weeks after that, I collect the tissue to study the acute toxicity and late toxicity effect. So this is from the acute toxicity cohort. I can see high dietary fiber can significantly increase the short-chain fatty acid level, including acetate, propionate, and butyrate. And also in this CRIP assay, so CRIPS is the basic function unit of the guts. It is very sensitive to radiation. It turns over every 3 to 5 days. As we can see, only in the very high dose of radiation, psyllium plus resistant starch and inulin can increase the remaining CRIPS, which means that this too high fiber diet can protect the guts from high dose radiation. And also, this is the body weight change after ionizing radiation, 0.2% cellulose, psyllium plus resistant starch, and inulin. As we can see, the dots line are ionizing radiation group. So the mice in this group experience a delayed weight gain compared to their non-IR control, but this was not seen in the other high fiber diets group. So which means that psyllium diet, especially psyllium plus resistant starch and inulin, can protect the guts, especially during the acute early stage. So in the end, I've also found that I also performed a couple of in vitro assays because Bacteroides acetylfascins is a potential radiosensitizer found in my previous project, master project. And then so that's why I try to use the bacterial supernatant of this bacteria and treat them on RT112 human bladder cancer cells. So this is the result. This is just a single bacterial supernatant, and then BA plus FP is the potential radiosensitizer plus short-chain fatty acid producing bacteria. As you can see, if I combine these two bacteria together, after 2 gray ionizing radiation, it can cause a significantly higher level of DNA damage compared to the other 2 single bacterial supernatant, which means that Bacteroides acetylfascins plus butyrate producing bacteria can enhance the DNA damage response, which is very important for cytotoxic, which is very important for radiosensitivity. So actually, I also have the result, but because of the time limit, I didn't show it here. I also saw that BA plus FP can increase the radiosensitivity in human bladder cancer cells, even in a dose-dependent manner. So finally, my conclusion is Bacteroides acetylfascins is a potential radiosensitizer, especially combined with butyrate-producing bacteria, fecal bacteria prognosy. And our in vivo experiment suggested that psyllium plus inulin and resistant starch can achieve tumor control, better tumor control combined with ionizing radiation. And it involved enhancement of short-chain fatty acid and also tumor immune response. So actually, there are still a lot of work to do. For example, oral gavage of this bacteria to prove the causal relationship effect between guts microbiota and the radiosensitization, or to find out what is the key metabolites produced by psyllium plus resistant starch in radiosensitization. So there's still a lot of work to do. So that's why microbiota gazing actually is still going on. So finally, I want to say thanks for all the day-to-day help from my colleague, especially Professor NQT, who is my supervisor, for being very, very supportive of my DPhil project. Thank you very much. Thank you.
Thank you. Any questions? While we're waiting for the microphone to go there, Chi, is there any evidence in humans that the microbiota might be important, or is this going to be a mouse-only phenomenon?
So actually, the very first study about guts microbiota and anti-cancer treatment is immunotherapy. So there are several papers have already proved that guts microbiota in human is related with the efficacy of immunotherapy. And then there is there are so many papers about that. And recently, with the advanced development of guts microbiota, we are quite sure that although the guts microbiota, especially species, are different between human and mice, but their physiological outcome are very similar because both of them need short-chain fatty acid, acetate, propionate, and butyrate for the guts health as the primary energy source for the guts. So the species might be different, but the outcome should be similar. Yes.
Cool. Thank you. Really nice talk. I just wanted to ask a bit about the sort of the increase in T cell numbers. So cancers that have got an FGFR3 activating mutation are typically immune deserts. Do you think this would be able to overcome that, or do you consider this might be an approach that would help basal tumors more than the more luminal or non-muscle invasive tumors?
Sorry, can you repeat again? FG?
So bladder cancers that have got an FGFR3 activating mutation, typically they have very, very low immune cell content. Do you think that this approach of a treatment approach would be enough to overcome that immune desert to allow the T cells in to help clear?
I'm not sure, but I think that is possible because when I was doing all the mouse experiment, I found that there are a cohort of immune cold tumor and there are a cohort of immune hot tumor. And it seems like our dietary fiber, if combined with ionizing radiation, not only the diet alone, it seems like if combining with the ionizing radiation, which can enhance the antigen presentation, it seems like there is a chance to turn the immune hot to immune cold to immune hot. So I'm not sure it's need experiment to prove that, but I think that's possible. Yeah.
Thank you very much, Chi.
Thank you.
So our final presentation today is from Benjamin Murray, who's going to tell us about an in vitro bladder cancer model which could be used as a test bed for novel treatments.
So hi there, I'm Ben, and I just want to say thank you to the organisers for allowing me to speak today. For a third-year PhD student, I'm really honoured to talk about my work. So one of the outcomes, the outcomes for bladder cancer have not improved in the last 40 years, and the patient-derived xenograft mouse model is the gold standard model and has yielded important insights. However, there are species-specific differences in the cell microenvironment between mouse and human bladders. So therefore, developing new human-based in vitro models to complement the mouse model could help further inform new treatments. So the aim for my project is to engineer a new in vitro human bladder cancer hybrid to be used as a test bed for potential new treatments. So I just want to introduce you to the H-Black urothelial model. So it was previously published from Horsley et al. In 2018 and has since been improved. So the H-Black urothelium is cultured for 18 days in 100% urine on the apical side until mature. It has 7 layers of cells and also differentiation of umbrella cells. It's grown on a planar transwell surface and also expresses the GAG layer and appropriate biomarkers CK8, CK20, UP3, and Z01. And it also has good barrier function. Our lab group are also using this organoid model in terms of research with UTIs. So just to quickly go through my methodology. So on the right-hand side is where we grow the H-Black urothelial organoid in transwells in 100% urine in the apical chamber and a calcium-rich media in the bottom. And so on the other side, I'm growing RT112 cancer cells, either tagged with EGFP LifeAct or dyed with a cell tracker green dye. And I'm making these into microspheroids in a microspheroid plate, which took a lot of work to optimize the growth conditions. And all I'm doing is I'm implementing the spheroids onto the H-Black urothelial organoid to make the H-Black urothelial cancer hybrid. So one of the issues for this model was the visualization. So at one point, I was growing very big spheroids, and so we had to use light sheet microscopy, and it looked like I was imaging a beach ball on a sheet of paper, basically. So in terms of getting the right detail and showing the right the detail for the model, the light sheet microscopy wasn't the best idea. So we then went into making smaller spheroids and trying to get the detail right. So we went for the confocal microscopy in the end. So there's a phenomena in bladder cancer where more invasive bladder tumors go through coherent switching. As we have only the urothelium, we can only say this is a model for TA grade bladder cancer. So in TA grade bladder cancer, E-cadherin is expressed in the tumor and the urothelium of TA grade bladder cancer. So, oh, there's the pointer, cool. So over here, we have the cancer spheroid over there, and also the differentiated urothelial layer, which all do express E-cadherin all at the tight junctions of the tumor and the urothelium. So this is quite nice to see. So it's expressed in tight junctions of cells of the hybrid model. So conversely, peak adherent, we wanted to see if it was weakly expressed, as it is only a TA grade bladder cancer model. And we can only see peak adherent is only really expressed in the cytoplasm of the tumor cells of the cancer spheroid, but also a little bit in the urothelium as well. So in terms of the cadherin expression, this was also shown in the hybrid model, which is nice to see. So another marker I wanted to see was the integrin alpha-3 beta-1 invasion marker. So this has recently been published to express on the tip of the filopodia of invading cancer cells, especially in mouse models. So I wanted to see if this was replicated in the hybrid model. So as you can see here, there are two cancer cells that have migrated from the main tumor spheroid. However, due to resolution and my technical ability as a PhD student at the moment, I can't figure out if the invasion marker is expressed at the filopodia of the cells. So what I have been asking at my poster as well at P4 is whether there is another invasion marker that I can use. So I'd love to get some help on that at the moment. So part of my aim is also to make for this hybrid model is to be a test bed for new treatments. And so we previously, as a lab group, we previously published a prototype called Capvaran. And this is a UTI treatment. So we've encapsulated UTI antibiotic nitrofurantoin in a PLGA polymer. And we've shown that it has good penetration of cargo into the H-flat urothelium, as you can see in this video here. So the capsules here in this video contain FITC dye, and when the capsules are put onto the H-flat urothelium, we can see the cells all go green inside to show the release of cargo. So therefore, using this same encapsulation technology, we want to enhance drug delivery of a chemotherapeutic. One of the main candidates we have for this one is mitomycin C, as we can instill mitomycin C at room temperature with these capsules. So just a preliminary experiment just to see if the capsules do penetrate into the cancer cells. Just did a 2D penetration assay of the capsules and found that actually the capsules do unload the cargo, which was Nile red dye, into the cancer cells here, which was a nice preliminary result. So at the moment, that's all the work I've done so far, but there's loads of potential with this model. So basically, I want to continue characterizing the hybrid model, looking at KI proliferation marker, KI67. Also incorporating a laminar propria layer to model for, so we can say we can model for non-muscle invasive bladder cancer. So I've got a student who I'm supervising that's going to help with that. We also want to incorporate other bladder cancer cell lines to make sure to validate the concept that we have with the hybrid model. So we want to use a high invasive T24 cell line, but also in the future, we could look at patient tumor cells for more personalized treatment and screening of other treatments. We also want to validate the model using histopathology. So we're in collaboration with a consultant urologist who will help advise whether it actually looks like a tumor or bladder tumor from a patient, which will be nice. But also, the exciting thing is continue the therapy testing, and also probably finishing with the testing with the encapsulated chemotherapeutic on the hybrid model. But also, in collaboration as well, I'm testing oncolytic viruses as another treatment method to hopefully validate this as a test bed for new treatments. So I just want to say thank you for listening to my talk. These are all my lab group who have helped with my work, and all the list of collaborators and my thesis committee. And if you have any more questions, and I'm sure you probably do, I'm at poster 4 if you'd like to talk to me. So thank you very much.
Thanks, Benjamin. Any questions?
Hi, thank you. That's a very nice talk. I just wanted to ask, have you thought about the possibility of incorporating the immune component as well, as we all know that for treatment of bladder cancer, especially non-muscle
Yes,
invasive?
absolutely. So one of the ideas we had was incorporating tumor-associated macrophages, but we think that in the scope of my PhD, I doubt I'll probably get to that. There is, as you can see, there was a lot more work we could do for that, but it was definitely on the list of ideas that we could do with this. So in terms of potential, again, I think the message I want to really convey is the potential with this model that we can do other things, and actually it could put more research out there, really, with in vitro cell-based models.
Thank you.
Thanks.
Any other questions? While we waited for the microphone to go there, Benjamin, how do you see your model fitting into the workflow from preclinical to phase 1 clinical trials? So obviously the idea here hopefully would be to spare in vivo modeling, but how do you see that working?
I mean, at the moment, so obviously mouse models they're very important at the moment, and they're a lot better than this model at the moment. So we really want this model to complement at the moment. So really complement the mouse model. So, I mean, 2 models validating 1 treatment is probably better than just 1 model. And as because of the species-specific differences, the human-based model could also inform other more information from new treatments. So getting it past into model treatments, I mean, would like it to be on a level with animal in vivo studies, hopefully.
Great, that was a brilliant talk. It's just really a comment. One of my colleagues here, Dr. Kelly Ward, she is poster P6. Actually, it's in fact, you guys are working on something very similar, and I thought it would be intriguing if you would talk to each other.
Absolutely,
go ahead.
yes, absolutely. No, thank you very
Sorry,
much.
Kelly, to put you on the spot.
Awesome.
Thank you very much. Thank you very much to all our speakers. I think we can break for tea a little bit early. Thank you very much.